Everyone. Thanks for coming to seminar. So today I'm really excited to introduce Dr. Kim Komatsu, who's coming to us from the University of North Carolina, Greensboro, where she's an associate professor. She's the Florence Schaefer Distinguished Scholar of Science and also the associate department chair. Before she moved there 3 years ago, she was leading a group at the Smithsonian Environmental Research Center in Maryland. Her research is really exciting. Most of her research examines how global change drivers affect community dynamics and ecosystem functioning, often focusing on species interactions like mutualisms. Her work also uses a lot of data and experiments from long-term experiments and collaborative experiments to synthesize the effects of global change across space and time. She's also really a leader in this area of long-term and collaborative research. She's on the Science Steering Committee at Konza Prairie LTER and is also on the advisory board of the Nutrient Network. So I'm really excited to hear about her research today, so please join me in welcoming her.
All right, can everybody hear me okay? Great, yeah, thank you so much for that really nice introduction. As Mia said, I think very broadly about a lot of topics in ecology. I think about legume-rhizobial mutualisms and plant-insect interactions and how plant communities are responding to global change. Most of that work I'm not going to have time to talk about today, but I've had some great conversations today with people about these various topics, and I'd love to talk more with anyone who's interested. But for today, I want to focus on the data synthesis work that me and a group of collaborators have been working on for the past 15 years now. So we've made a lot of progress, and as you'll see, we have lots of room to grow from this work as well. And we're really focusing here in on how we can use plant traits to tell us something about how plant communities are going to be responding to global change. So that's our topic for the day. And I just want to start out by sharing with you all my love of my study system, which are grassland ecosystems. These are the systems that we're using in our synthesis work, so you're going to hear about them for the next hour. So hopefully I can convince you all that for at least the next hour, grasslands are important to think about. So I love grasslands for lots of different reasons. The first is that they're just absolutely beautiful landscapes, in my opinion. They have these big, beautiful skies and the verdant green grass below them. We sing about them in our most patriotic songs, right? Like our amber waves of grain and our spacious skies. So there's lots of people who find beauty in this system. Grasslands are also thought to be a driver of human evolution, which I find fascinating. So the rise of the lineages that led to modern humans are thought to have co-evolved with the C4 grasslands that emerged in Africa. And traits like bipedalism and our complex social structures are all thought to be caused by our close interactions with these grassland systems throughout our evolution. So that's a fun fact. But then, of course, they're also ecologically really important, right? So they cover 40% of Earth's ice-free terrestrial surface. They're home to a wide variety of plant and animal diversity, much of which is endemic to these ecosystem types. They provide really critical ecosystem functions and services to humans, so things like nutrient cycling and water filtration, erosion control, carbon storage and sequestration below ground are all critical services provided by these systems. They're highly tractable systems, which is great for the work that we're doing. So they're easy to manipulate. They're relatively easy to measure the responses. And a lot of our fundamental theories in ecology have come out of grassland systems. So I think they're important to consider for that in that respect as well. And then, of course, our grassland systems, like many ecosystem types, are highly endangered around the world as well. So this is just a stat from North America that 40% of our grasslands currently remain compared to their historical extent. So we've lost about half of our grasslands. But the ones that remain, only 2% of them are intact. So most of them are degraded systems as well. And so we really have to think about how can we conserve these ecosystems, restore these ecosystems, and protect them going forward too. So that's my soapbox about grasslands, and I will stop there. Hopefully, again, you're convinced to think about them at least for the next hour. And I want to talk to you today about how our grassland communities are changing over time. So like many different ecosystems, we see plant community change through time through successional processes. We see plant community change in response to changes in environmental drivers. And potentially we're seeing changes as well in response to global change. So we know human activities are driving big changes in Earth's biogeochemical cycles and our climate cycles. This figure here is showing, if I can use the pointer, across the x-axis here, we're looking through time, and we're seeing an increase in the global human population. That's that blue line there. And simultaneously, we're seeing increases in carbon emissions, leading to increased atmospheric CO2, increased total reactive nitrogen, changes in the global temperature anomaly. So we're really drastically changing our ecosystems around the world. So that's unfortunate, but fortunately, I would say we're also in an era of global change experimentation. So we have been spending the past several decades imposing experimental global change drivers in our grassland communities and trying to figure out how these communities are going to be responding over time and make predictions about what we should be seeing in the future. So there's all kinds of different experiments looking at all kinds of different global change drivers in a wide variety of different methods. And so this is great that we're gathering all this data, but some of our data can lead to confusing results, right? So here I'm showing you data from a single field site, the field site that I do most of my research at, the Konza Prairie LTER, which is a tallgrass prairie site in eastern Kansas. So just, yeah, a little southwest of here. And at this site, we do a lot of long-term experiments. And this figure is showing data across 6 different long-term experiments that have all been set up independently, but they're all manipulating nitrogen availability in the soil. So we're adding relatively high levels of nitrogen at about 10 grams per meter squared per year over at least a decade for each of these experiments, in some cases many, many decades. And so in this figure, we're looking at the 6 different experiments are shown as the different colors. And this is a multivariate plot. So we're looking at the entire plant community represented by a single dot here, representing species abundances and their identities encompassed within that one dot. And the further away the dots are from each other, the more dissimilar the plant communities are from each other. And so when we're looking here, we're looking at the starting year of an experiment at the head of an arrow, and the final year of that experiment and our nutrient addition plots at the end of the arrow. And we can see, even at the same experimental site, same experimental treatments, we have very different starting plant communities and very different ending plant communities within this space. And I'll just draw attention here to these 2 teal and darker teal dots at the top. These 2 experiments, the nut-net experiment and invert experiment, were set up in the same watershed at Konza, so they're about 20 meters away from each other, 1 year apart. One was started in 2008, one was started in 2009. They started with relatively similar plant communities, which is great, what you would expect, but they've ended up in very different parts of this NMDS plot, right? So even co-located experiments started at relatively the same time, we're seeing very different responses. So this leads to the question, can we generalize how plant communities are responding to global change? If we can't even do it at a single site, how can we do this globally, right? And so together, myself and my colleague Megan Avolio from Johns Hopkins University, back when we were graduate students, actually set out to address this question by gathering data from a bunch of different experiments around the world and trying to come up with these synthetic principles of how plant communities are changing to these global change drivers. And so I asked Megan, can you send me a recent picture of yourself? She has like a fun new haircut. I wanted to have a recent photo, and this is what she sent me. So that gives you a little indication of Megan's personality. So here's an old photo from like 10 years ago of Megan. But yeah, so this is work that we started as graduate students. Megan's one of my best friends and close community ecology partners. And none of this work would be possible without this collaborative effort. So together, Megan and I applied for funding from the LTER network to start this, what we call the CoRRE database, or CoRRE working group. So we're looking at plant community responses to resource experiments. And we gathered together a great team of scientists to do this work. At the time, most of us were either graduate students or postdocs, and we've all now found success as faculty members at various institutions across the country, which is great. And since that time, we've had 2 more iterations of this working group building on the work that we've been doing with funding from NCs and IDIV, so we've really expanded our collaborations globally. Lots of different people have been involved in this work, and again, everything I'm presenting to you is because of these great collaborations. So none of this is done alone by me. Okay, so the CoRRE database are a database of experiments, 134 experiments from 64 sites around the world, where we collected data from individual investigators who had set up their own experiments. And our requirements were that those experiments had to have directly manipulated a resource. So they had to have manipulated either soil nutrients, water availability, light availability, or atmospheric CO2. They had to be in our tractable herbaceous ecosystems. They had to have at least 5 replicates so we can look at variability in the responses. And then they had to be at least 3 years long so we could ensure that there was some sort of response. And so you could see the distribution of these sites. Many of these sites, of course, they're making measurements over time, so we have lots of different time points and species occurrences across this database. And so then we asked our question, can we generalize our plant community responses to these global change drivers? And to do this, we used, first, a really commonly used metric of plant community responses, which is plant species richness. So just the number of species that you can count up within a plot. And so here in this figure, we're looking across the standardized year of the experiment on the x-axis. So this is how long an experimental treatment has been chronically imposed in these experiments. And then the y-axis is the proportional difference in plant species richness between our treated plots and our control plots. So positive numbers means that the treatment plots are gaining species relative to the control. Negative numbers means that the treatment plots are losing species relative to the control. And then each of these individual gray lines here are the individual treatment responses across all of those 134 experiments around the world. And then this thick black line down the middle that looks like the x-axis is actually the global mean. So what's the overall response across all of these experiments around the world? And as you can see, we have a very flat line here. So not only is our global mean right around 0, we're not seeing plant richness responses to our global change treatments, but across all of these individual gray lines, 78% of the time, we are not seeing richness differences in our plant communities in response to these experimental global change drivers. So we can say here that plant species richness seems like it's probably not a very sensitive metric to global change. But when we look at our overall community differences in that multivariate sense again, thinking about plant species identities and their abundances, we do see a big response here. So our global mean, again, is this thick black line, and we're seeing through time increasing plant community change in our treatment plots relative to our control plots. But again, there's huge variation if you look across these individual gray lines. Some of them are totally flat with no response. Some of them have linear increases. Some of them start to change and then revert back towards the control communities. So again, we're not able to generalize what's happening across all of these different experiments in a really satisfying or robust way. So we can say here that community responses to these global change drivers are really dynamic, and we need to have more information to be able to figure out what's happening at each of these individual sites. So thinking about how we can get closer to these generalizations, back in the 1990s and early 2000s, people were really positing that plant traits should be this holy grail of how we could understand what species should be succeeding with global change, what species are losing, and what that means for the functioning of our ecosystems, right? And so this is measuring things, the easy things, which I've had some good discussions with some of the graduate students about what traits should we be measuring. So we've measured the easy things like plant height, or the thickness of a leaf, or the area of the leaf relative to its weight, right? So all of these kind of abstract measurements that we think maybe could say something about how these plants should be responding to global change. And so these are our questions for today. Can we use plant traits to understand these mechanisms of species co-occurrence at our sites? What traits make species winners or losers under these global change conditions? And then what are the consequences of global change for overall community functional diversity? All right, so to answer these questions, of course, we have to have trait data. And so we gathered this trait data for 6,211 species that we found across the CoRRE database, but also across 2 additional databases that I'll talk to you about in a little bit. And we wanted to look at many more traits than this, but these are the ones we settled on. We have 6 categorical traits, so thinking about the plant's growth forms, their photosynthetic pathways, their lifespan. Are they clonal in their reproductive strategies? Are they associating with microbial mutualists like mycorrhizae or nitrogen fixers? And then also their continuous traits. So how tall do they get? What's the specific leaf area, leaf dry matter content, leaf nitrogen, specific root length, and seed dry mass. So all indicators of the acquisitiveness or conservativeness of these species. And so I just want to take a little detour here and talk about trait data. So when we were trying to collect this trait data for these 6,000 species, we went to the normal sources. We looked across a bunch of different plant trait databases, including the TRI database, which is the most commonly used database that we think about as plant community ecologists. And what we found is for the vast majority of our continuous traits and almost all of our categorical traits, the trait data was super sparse. So for any given trait, we here at the high end, we maybe had between 18 to 19% of our species covered by these databases in terms of their plant height or leaf dry matter content, which means we have 80% missing data for the most common traits that we had in these databases. And then for most of the traits, we're down below 10% of our data is complete. And for things below ground, like our roots, it was almost nothing. We were missing 99% of the data. So the data were super sparse. And when you're doing these overall community analyses, you want complete trait data for every species in your community, right? You can't just think about the most common species or the most easy to measure trait. We also found across these databases, the trait data were really messy. So oftentimes there were errors in the data, which were easy to spot for the categorical traits. So like something could have been listed as a grass when we knew it was a forb, but how do you catch those errors for something like plant height, right? If there was a typo, you might not be able to figure that out. And then there was also a lot of repeated data that had been entered multiple times within the databases, so we had to clean all that data out so we weren't overrepresenting certain observations. So we have a whole pipeline that we went through to do this, collecting trait data from these 5 different databases. For our categorical trait data, we use these databases as a reference, but again, there was mostly missing data. So we did a pretty exhaustive literature search to fill in all of that trait data for those 6 categorical traits for every single plant species, which was a lot of human brain power going into this. And then for the continuous trait data, again, we were missing about 80% of our trait data here. And there's no way to go out and gather that information for all those species. So what we did is we turned to Bayesian hierarchical modeling to impute the data. So we used the relationships between these different trait variables, as well as the phylogenetic relationships of the plant species that we were looking at to computer generate our trait data for that 80% missing data. And our models did a really good job, so we had some training data and some validation data, and we were able to find really strong correlations between the imputed data and our validation data. So this was obviously a big effort, and I guess I should also say here, this trait data is now publicly available in the Environmental Data Initiative database, EDI, and so I'd be very happy to talk to anyone about it. We would really love people to use this data, and if you do use it and if you find mistakes, which we know there's probably mistakes, please come talk to us about it so we can fix them, so people can have confidence in it going forward as well. So that's our detour on traits. They're hard to come by, but good to use. And so now we can ask our questions now that we have our trait data in hand. Can we use plant traits to understand our mechanisms of species co-occurrence? And so to dive in here, I want to bring you out into one of my field sites. This is a site in western Kansas. It's a mixed grass prairie site where I would go out into the field, I bring my quadrat, I put it down in this grassland, and this is what I would see. So there's a big sea of plants here. And we have some relatively rare species. So here on the left, we have side oats grama, we have a white sage in the bottom and aster in the top. These are all relatively rare species in this community. When we're doing our plant richness counts, we are mostly counting these species, right? They make up the diversity of the plant community. But the vast majority of the plant biomass in this picture is made up of just 2 species. So we have prairie dropseed in the purple and little blue stem in the yellow here. So these are our co-dominant species in the system. They make up the vast majority of the abundance of plant cover, as well as the vast majority of the function in terms of productivity of the system. And so the term co-dominance has been relatively loosely used in the literature in the past. And so relatively recently, back in 2021, Jess Gray and Melinda Smith and I tried to come up with a more holistic definition of what co-dominance means by looking across all of these previous papers in the literature. And we came up with a pretty simple definition that co-dominant species should be those that have a shared abundance that together comprises the majority of total abundance in the community. And Jess came up with a clever metric thinking about how you can calculate the number of co-dominant species you should have in your system based on the harmonic means between the most abundant species and the next most abundant species within the system. So we're using that metric for our definition here today. And so we can say, within these ecosystems, do we have a single monodominant species? Do we have 2 or 3 co-dominating species? Or do we have a bunch of species that are relatively even in their abundances? Those are our 3 categories. Why should you care about co-dominant species? We could think that they might be important for something like ecosystem stability, right? So if species are trading off in space or in time, in their abundances or tolerances of environmental conditions, that could stabilize our functioning of our communities. They might be important for restoration efforts, right? So if you're trying to bring back your grassland plant communities, you have to think about what species should be there and what proportions. And for what I'm interested in, particularly today, is that they could also be really useful for us thinking about coexistence theory, right? How can these 2 species have really high shared abundance within this ecosystem, and how do they keep everybody else out? So that's our question for today. What filters influence species co-dominance? And to look at this, we use the CoRRE database that I've already described to you. We also use 2 additional databases. The Grazing Exclosure Consortium is an assembled database of experiments from around the world where people are manipulating grazer abundance. And then the Nutrient Network, I think many of you have heard of or talked to me about today, is this coordinated distributed experiment where people are using the same methods to look at nutrient availability and grazer presence and their influences on plant communities. So across these 3 databases, we have 527 experiments across more than 300 sites around the world. And when we look across all of these different sites, we see some interesting patterns pop out. So the vast majority of these sites are monodominated. About 60% of them have a single dominant species, and then a relatively rare subgroup of that makes up the diversity of the site. And you can see these are distributed pretty evenly across the globe. So those blue dots represent our monodominated communities. But co-dominance is not rare. About a quarter of the time, our sites are co-dominated by either 2 or 3 species that share that really high abundance and make up the majority of the functioning in the ecosystem. And again, these sites that are indicated by the red dots also seem to be pretty ubiquitously distributed across the globe. And so we wanted to know, we see co-dominance is relatively common. It occurs about a quarter of the time. Where is it occurring more across our planet? And so when we looked at a bunch of environmental factors, these are the 2 that stood out the most to us. So we're looking at mean annual precipitation on the left-hand side, mean annual temperature on the right-hand side, and then the probability of whether those sites would be monodominated in blue, co-dominated in red, or even communities in the yellow. And we see that at our extreme ends of precipitation, so our really high productivity sites, we have a swap over where you're more likely to be co-dominated than you are to be monodominated at these high levels of precipitation. We also see a similar trend for temperature, although the lines don't cross. So our warmer sites also are more likely to be co-dominated than our cooler sites. So overall, we can infer that these more high productivity sites are the ones that are more likely to have co-dominant species. So then we can ask what filters are driving co-dominance, co-occurrence across these sites. And we could think about this in a traditional community assembly kind of framework. So we might have our species pool across the top here represented by these fun little shapes. And those species can arrive to a site and they will be selected for based on their traits for whether they could withstand the environmental conditions at that site, right? This is our abiotic filter. So we might expect that environmental filtering could be promoting these co-dominant species with really high shared abundances that would have similar traits to each other because those traits are what allow them to do well under these environmental conditions. And we might expect this to be more common in our stressful environments. And then once species are able to tolerate those environmental conditions, then they start interacting with each other. So here we have our biotic filter that brings us down to whatever our final local community is. And once they start interacting with each other, we might expect that it would be better to have dissimilar traits from each other. So having trait trade-offs along these axes could promote co-dominance through niche differentiation, really making these species different from each other. And we might expect this to be more common in our resource-rich environments. And so we can test this looking at the functional distribution of our species. So this is a functional dendrogram here with our more similar trait species clustering together and species that have very dissimilar traits farther apart from each other in this dendrogram. And we can think about all possible pairwise combinations of species distances within this dendrogram. So we would have, again, some that are more similar, some that are more dissimilar, and a bunch in between. And we can array these values across a density plot of our trait dissimilarity. We would expect somewhat of a null or a bell curve distribution. And then we can ask for our co-dominant pair, how do they compare to everybody else in this species pool? So here we would think that if the co-dominant pair has more similar traits to each other than expected on average from this overall species pool, we might be seeing something that's indicative of environmental filtering. And on the flip side of that, if our co-dominant pair has much more dissimilar traits from each other, we're thinking this might have something to do with niche differentiation. And so we can look at these numbers. Again, now we're thinking about just that co-dominant pair compared to the null distribution of all possible combinations of species. So this is our relative deviation of functional distance on the x-axis, and a density plot across all of the sites that had co-dominant pairs. So we're ignoring all the monodominated sites, we're ignoring all the even sites, only thinking about those co-dominated sites. And we see a bimodal distribution here. So we see a strong peak on the left-hand side where those co-dominating species tend to have more similar traits than expected on average based on the overall community. And that would be indicative, again, of our environmental filtering. We also see another much smaller peak over on the other side where these species at those sites tend to have more dissimilar traits from each other than expected by chance. And so again, we're thinking this is indicative of that niche differentiation. So here we're seeing either environmental filtering in most of our cases, or niche differentiation in a substantial portion of cases could be promoting these co-dominant species coexisting at these high abundances. And that's all under control conditions. So what happens if we now start to impose our global change drivers on these plant communities? And we would expect that these altered environmental conditions should be shifting species abundances, either by altering the environmental filters, so changing moving species away from their tolerances environmentally, or through competitive exclusion, right? So we might expect to see a decrease in the number of codominant species because of these shifts away from their environmental tolerances or because of increased competition. And we would also expect to see shifts in that distribution of trait dissimilarity. So first, thinking, does the incidence of codominance shift with global change? So in this figure here, we're looking on the left-hand side at the incidence of our 3 different dominant syndromes in our control plots. So as I mentioned before, the majority of the time we see monodominance, a fair amount of time we see codominance, and slightly less we see even communities. And then how do those change when we impose our global change treatments? And we see, if anything, a slight increase in the incidence of codominance within our global change treated plots. But where those communities are coming from and where they're going seems to be all over the board and relatively proportional to their overall abundances in these different trait syndromes or dominant syndromes. So a substantial portion of the plots that were monodominated stay monodominated, but a large portion become codominated, some become even, and same for the codominance, right? There's a substantial subset that become monodominated, some stay codominated, some become even, and same for the even. So we haven't yet teased apart why some sites are doing, or some treatments are doing some transitions and others are not. But it's an interesting pattern to think about here. Okay, so then we can ask, are our filters altered with these global change treatments as well? So just to remind you here on the left-hand side, this is the figure I showed you before in our control plots, where we have that bimodal distribution. We have a relatively high peak on the side that we think is indicative of environmental filtering, more similar traits, and a smaller peak over on the dissimilar trait side, indicative of niche differentiation. And so when we impose our global change treatments that are stressful, we would think would be increasing environmental filtering. These are things like experimental warming or drought manipulations. We see a shift in this peak towards the left-hand side here. So species are becoming more similar in their traits to each other than we would expect by chance under these increased environmental stresses of our global change treatments. And the opposite is true when we impose global change treatments that are increasing resource availability. So increased nutrient availability, increased water availability. We're seeing a shift in our peak here towards a truly bimodal peak, where we have much more, many more pairs of codominant species that have more dissimilar traits from each other than would be expected by chance. So overall here, we're seeing that our abiotic and our biotic filters both seem to be maintaining codominance in our ecosystems. Our codominant species tend to be more functionally similar than we would expect by chance, but there are a subset that exhibit this trait divergence, which we think might be indicative of niche partitioning, and we're still teasing apart what are the environmental factors that are driving those dissimilarities across our sites. And then global change is shifting these dynamics, where increasing stress in our environments is increasing the strength of this environmental filter, while increasing resource availability is increasing competition and therefore niche differentiation among our codominant species. All right, so we have used our plant traits to try and understand these mechanisms of how species can be co-occurring in our ecosystems. And now we want to know, can we use these traits to make predictions about what species should be winners and what species should be losers under our global change conditions? So I'm going to talk to you about another metric here that you don't have to pay too much attention to the details, but here we're trying to think about the abundance and the frequency of species across our treatment and our control plots. So we can think of a species that might be locally really abundant in a certain control plot, but it's relatively sparsely present across most of our plots and at lower abundances in many of the plots, right? So this would be a species that potentially has a low mean abundance across our plots and a low frequency across our plots. We could think in our treated plots, maybe that same species might have really high abundance in many of the plots, and it's found everywhere, right? So it has high mean abundance and high frequency. And so then we can compare this, I'll call it the dominance index of a species in the treatment plots versus the control plots. and decide whether that species is either increasing in abundance and frequency with our global change treatments. That would be a positive difference in this dominance index. And that we would call those the winners, versus species that are decreasing in abundance and frequency with our global change treatments. These are our losers. And so we can ask what traits are making our species winners or losers with our global change conditions, right? And this is something that is not new. People have been thinking about this for a long time. So I'm showing a Grimes CSR triangle here, where there might be certain traits that lead to species being really good competitors in high resource environments, traits that make species successful under stressful environments or high disturbance environments. And there's been many decades, more than 50 years of work on this. This is a fun recent paper that came out. Looking within this CCA space, they can use a bunch of different plant traits to say these are the traits that make species good competitors, or these are the traits that make species good stress tolerators, right? And so we should be able to make predictions about specific trait syndromes that should be important under our various different global change treatments. So for example, in our nutrient addition treatments, we should have resource acquisitive species, those that have high specific leaf area, those that can grow really tall. In high disturbance areas, we should see species with lower seed mass so they could immigrate into the area after disturbance. In warmed environments, maybe we would see more C4 or CAM plants, so these species that have high water use efficiency. And of course, you could use many more traits to make many more predictions across our different global change treatments. And so we tried to do this, looking across our different global change treatments that are imposed across our different experiments around the world. So each of these bars, different colors represent our different global change treatments. And then we're looking at whether a plant species would be considered a winner or a loser based on whether they have that plant trait. So if they fall on the left-hand side of this figure here, they would be a loser if they have that plant trait with that global change treatment. If they fall on the right-hand side of the graph, they would be a winner with that global change treatment with that trait. And then the different panels represent our different traits. So we can just do a basic gut check here of does this work? Thinking about our categorical traits that are a little more intuitive to understand. So here we're looking at, on the top, our C4 and CAM plants tend to decline or be losers when we apply irrigation treatments. Again, this makes sense because these plants are much more water use efficient in their photosynthetic pathways. So you would expect if you're applying a lot of water to a plot, they're going to be losers, right? They're losing their competitive edge. Similarly, our nitrogen fixers, which are getting nitrogen out of the atmosphere, tend to be losers when we add nitrogen to the soil, right? They're losing that competitive edge. Other species are able to outcompete them. So these trends make sense. And then we could start to think about some more complicated trends or more abstract ideas with our plant traits. So thinking about, again, our resource acquisitive traits, species that have high leaf nitrogen or high specific leaf area on the top panel here and the bottom panel here, they both tend to increase in abundance when you add nitrogen or when you add multiple different nutrients simultaneously. And species that have resource conservative traits like high leaf dry matter content are ones that tend to be losers when we add these nutrient additions. So again, intuitive results that we can combine together to say what are the trait syndromes that make these species winners and losers under our global change treatments. So overall, it seems like we can predict our winners and losers based on their traits with these global change treatments. And of course, there's many more traits that I didn't show you here, but we're writing up this paper, and hopefully you can check it out relatively soon to see more of these results. All right. So to round it out here, thinking about how now our global change treatments are influencing the overall functional diversity of our plant communities on the whole. So not just thinking about the dominant species or the co-dominant species, but all of the members of our community. So again, we could think about the functional diversity of our plot thinking about all of the different species that are present at different abundances, right? So we have some common species and some rare species. And we would think that the functional diversity or the trait dissimilarity across all of these species should be relatively high with high resource availability. Again, building off those predictions from before that you would have more niche differentiation under more competitive environments. And similarly, we would think that the functional diversity should be lower under high environmental stress conditions, right? So you're filtering species for their environmental tolerances, and you might expect them to have shared traits because of those tolerances. And so again, we can synthesize across all of our different data sets, looking at the effect sizes we use the Rao-Q metric of functional diversity. So thinking here, these are our mean responses across all of our different global change treatments or experiments for this particular global change treatment with 95% confidence intervals around them. So if our line is crossing 0 here, that means we're not seeing a response. And so our response to nitrogen, which would be a high resource environment, we actually see the opposite of our prediction, that we see a decrease in functional diversity when we're adding nitrogen across these different sites around the world. And I'll just show you one example of what this might look like from the site that I know the best, the Konza Prairie site. One of those 6 nutrient addition experiments I told you about at the very beginning of the talk is the change experiment, where we've been adding nitrogen across a gradient of levels for the past 10 years, 13 years now, actually, and watching the plant community response. And I'll just show you the response in the control and the 10 grams per meter square of nitrogen plots. So here in this PCA space, we're looking at each dot representing an individual plant species sized by its relative abundance. So the bigger the dot, the more abundant that species is. And then the arrows are showing our trait loadings within this space. And I only labeled the more dominant traits that were driving these responses. So in this system, we have a dominant species, Andropogon gerardii. This is our big blue stem. C4 clonal grass grows relatively quickly in these environments. And it's also really efficient in its nitrogen use, right? It can draw down nitrogen to relatively low levels in these plots. And then our other co-dominating species in these plots under control conditions is Lespedeza capitata, our bush clover, which is a nitrogen fixer. It's a forb, and it also has relatively high leaf nitrogen because of its mutualistic association with rhizobial bacteria. So these are our 2 co-dominant species in these plots under our control conditions. So now we can layer on what happens to this trait space with our 10 grams per meter squared of nitrogen addition. And we see a variety of changes. So in some cases, we see species disappearing from the plot. So for example, these species go away. Some species up here. So for example, this one pops in. But these are all relatively rare species. They're not making up the majority of our functional trait distribution here, which I forgot to mention we used an abundance-weighted metric. But what we do see is that one of our 2 co-dominant species, that Lespedeza, shrinks down to very low abundance, right? So we're seeing a loss of our nitrogen fixer in the system. It makes a lot of sense that that would happen when you add nitrogen. And the abundance of our C4 grass increases slightly. So we have slightly more of this dominant C4 grass to become the monodominant species in this system. But we're also seeing relatively large increases in the abundances of a few other C4 grass species that's shrinking this functional trait distribution, right? So this is why we're seeing this decrease in our overall functional diversity at this site. And this is just one example of one pattern at one site. We've looked at similar PCA diagrams for a bunch of different experimental treatments at a bunch of different sites, and this decrease in functional diversity could happen in any possible way that you could imagine. So again, there's generality in the response, but the mechanism underlying that response seems to be really different across these grassland sites. So room to explore. But when we look at our other high resource treatments, addition of phosphorus, irrigation, so water addition, or increased atmospheric CO2, we're not seeing big changes in our functional diversity response at the community level. And then similarly for our high stress treatments, the majority of the time we're not seeing responses to things like drought or warming or disturbance. But we do see a decrease in functional diversity when we remove herbivores that should be present in a system. So often you think that you're reducing light availability, reducing disturbance in that system, as well as when we have multiple compounding global change factors all applied at the same time. So on the whole, it seems like these herbaceous ecosystems tend to be fairly functionally robust in terms of their functional trait diversity to a lot of these global change factors that we're imposing around the world. But when they are impacted, it seems like we're losing functional diversity, we're not gaining it. And so we can imagine that these shifts in functional diversity should have consequences for the stability of our ecosystems, right? If you're losing functional diversity, potentially you're going to have more unstable functioning in the future. All right, so wrapping it up here, again, we've synthesized across hundreds of different experiments around the world, and we do find some broad generalizable patterns, which is exciting. So we see plant communities are changing, of course, but plant species richness maybe is not the best metric to use to capture these changes. We can use our plant traits to understand what the mechanisms are that allow species to co-occur at really high abundances, and how those are shifting under global change treatments. We can use our plant traits to predict who's going to win and who's going to lose under these global change treatments, and ultimately what that means for the functional diversity of the system, right? And so I'll just end here by saying that our next step is actually kind of going backwards in time. So our second iteration of this working group was bringing together plant community ecologists and field empiricists who are out there gathering the trait data, collecting our plant community data. We're really interested in the nuances of the mechanisms of what's happening in the field, linking our traits to our community structure and our ultimate ecosystem function. And then a bunch of process-based modelers who are trying to incorporate this information into these big regional models to say something about how the world is going to function in the future. And when we first started these conversations, they were relatively challenging. We were using very dissimilar terminology. The process-based modelers told us that they don't actually ever think about grass, and if they do, it's modeled as a carpet of slime, which is like hurtful to hear, but we're thinking about very different processes at these two different scales, right? So they're thinking a lot about fluxes of gases and broad functional types of plants. So like, are you a tree or are you a grass, right? Are you in the carpet of slime or are you one of the few functional groups of trees that they actually care about? Whereas we're thinking about these individualistic species responses, how these species are coexisting together, all of the trophic dynamics that are occurring, most of which don't go into these big ecosystem models. And so we're kind of circling back to this work to now try and bridge that gap in both directions. So can we incorporate some of these traits and these factors that we think are important as community ecologists into these big models, and vice versa? Can we be measuring the traits and the fluxes that the process-based modelers think are important to think about on the ground across these different global change experiments? So there's a lot of room to grow here, and we hope to keep working on this for the next 15 years as well. So with that, I'll stop there. And thank all of our data contributors and the people that have worked on all of these analyses. Again, none of this is possible without hundreds of people who have contributed here, as well as our funding sources. And if you want to learn more, we have a website for the CoRRE database as well that has many other results and publications that I didn't have time to talk about today. And I'd be happy to take any questions.
Thanks for that talk. Uh-oh, is it on? All right, well,
I can hear you.
assume it's on. Thanks for the talk. It was great. So I have a question. And maybe I missed it, but most of what you're talking about is abundance. Is that correct?
Yeah, so we're thinking about the abundances of the different species within the communities, and then the traits that those species have that we can link to those abundances. Mm-hmm.
So how might your results change if you thought about biomass instead of abundance?
Yes, that's a good question. And the way people have measured species abundance across these experiments is very different. Of course, these are all individual experiments that investigators have set up. We're trusting them to do whatever's right for their system. So some of them actually are measuring biomass, species-specific biomass within the system. That's relatively rare, and there's flaws with that, right? Because they're moving where they're clipping so that they don't have a grazing effect, so you're not getting your static plant community through time. Most of the people are looking at cover data, so estimating percent abundances. Some people are looking at pin hits, and a few other methods that are much more rare. So this is a good question. I would say that many grassland people would argue that cover and biomass are tightly correlated enough that you would see similar responses. I don't know if I fully buy into that argument, and I think potentially there could be big differences, especially if you have maybe one individual. If you're thinking about densities versus percent cover versus biomass, you could have a large individual that's a single individual plant species, or plant individual. How many times can I say individual in the same sentence? But it makes up the majority of the biomass in a plot, right? And you would maybe expect different patterns if you had that kind of scenario. But in many of the cases, I do think there's a pretty tight correlation between a species abundance by percent cover and its biomass in the plot.
Cool, thanks.
Oh.
It's definitely not working now. As you know, a lot of people have made their entire careers where they manipulate species diversity in plant communities and look at how that changes ecosystem function, and they argue that we're losing species. But your work and that of many others shows that we're not at those scales. And so I wondered what you thought about all those experiments.
Yeah, I think this
and
is...
I've checked, no one in the room has done those experiments.
Yeah, I was going to say, who's my audience here? No, so this is actually part of the impetus of this work was thinking about what we see down at Konza Prairie, where we think a lot about dominance and co-dominant species, and that they're the main drivers of our ecosystem function by definition, versus the work of our colleagues up at Cedar Creek, which have the same species. They're also a tallgrass prairie ecosystem, but they think a lot about biodiversity ecosystem function and the richness side of things, right? And so when we first set up this working group, we brought the people who work at Cedar Creek to the working group as well to try and come up with these more holistic understandings of what's happening. And I would say that even those people who think a lot about biodiversity ecosystem function have to admit that richness is maybe not the best metric to look at community responses to global change, right? So oftentimes we see conflicting patterns or no response at all. But that doesn't mean that those rare species aren't important in their own right. I would again still argue that the definition of dominant species means that they're the ones contributing most to the functioning in the ecosystem. But those rare species are also contributing to functioning and stability of the system, certainly. And so we should, when we're thinking about global change and how it's impacting our communities and the functioning of our systems, we should certainly consider all the species that are present. But maybe the dominant species a little bit more.
Any others?
So in the beginning, you showed your really recent paper where you had all these like nitrogen experiments from Konza Prairie, and even the ones that like had really similar starting communities ended up being really different. I'm wondering if like after seeing that, has that changed the way you think of like nutrient network experiments or other like comparing across distributed experiments?
Yeah, totally. So I think there's so much value in the nutrient network where you're using the same experimental treatments at all these sites around the world. So you're really controlling for methodology, which our 6 experiments did not do at Konza Prairie. They're very similar, but they're not exactly the same. And so that can help you say something about, it's not methodological differences to the extent that we could control for that. But there's something about these plant community responses and how they're truly changing in response to these treatments. And I will say there's, of course, a lot more nuance to that paper as well, where we see very consistent timelines of change. So all of these experiments, it took about 6 years where there was pretty consistent plant community change over time in whatever direction they were going, before we saw like this ultimate community that the plots stabilized around. So there were consistencies, but then also it seemed like there were a lot of potentially priority effects where you had a resource acquisitive species that was present at relatively low levels at the start of an experiment, and if it happened to be in these plots within an experiment, it was the one that took over. And this other experiment over here might have had a different species that was present at low levels, and it was the one that took over. So again, I think also going back to the previous question, thinking about those rare species could really matter with global change because they're the ones that are more likely to become dominant under these altered conditions. But yeah, that's a long-winded way of saying I think these coordinated networks are really valuable in terms of the vast amount of data that they've provided and the consistent way that they've provided it, for sure. And one of those experiments was a nut-net site, which is fun. So we could compare our responses to the rest of the network.
And then let's thank Kim again.
Thank you.